Okay, so what this means is that ACE is the enzyme, always an enzyme, means does reactions. Isomer, if you recall, means the same thing. An isomer is like, it's the same parts to it. So this is just telling you that this is not changing the parts. So it's an enzyme that doesn't change the parts, but it changes the shape, the topology. As in, it cuts the DNA and lets it unwind and then puts it back together. Not changing anything, just keeping it from making a shape that's going to break. That's how I remember that name. It's just changing the shape of the DNA, the winding, but it's not actually changing any, it's not doing any reactions to where it's adding anything or taking it away. So topoisomerase is going to sit in front of here, so when the DNA opens, it doesn't get overcoiled and break. Helicase is right at the fork behind it, opening the fork, so it's moving that way. On the other side, helicase will be moving the other way. What else do we need? We have the DNA builder, which is the DNA polymerase, always adding on to the 3'. How do we know that's 3'? Because it has to match opposite with the 5'. We have our primase that built the primer first. Why? Because DNA polymerase, just because of its shape, can't add on to naked DNA. It can only add on to OHs. So you gotta like make an OH first. And what else do we have? So DNA, once it's open, the tendency is for it to close back up. Those hydrogen bonds, they just form. Just like water sticks to water. So in order to keep the fork from like reclosing on itself over and over, little proteins sit on the DNA and basically block the DNA from reclosing. So these are called single strand. That makes sense. Binding proteins. So we've got all these little single strand binding proteins, which are just keeping the DNA helix from closing. That's good. They get bumped off as polymerase comes. This side, that's all you need on the leading side. The lagging side is the one that's built backwards, and so that's tricky. So the first primer would go here because the DNA opened. It opens here, and it's it's opening and then opening like this, right? So closed. You got the origin of replication, and then eventually it just opens up. So here's where your DNA started. Because the 3', we know that matches with the 5,
yep.
Which means this is the 3, which means there's the first primer. Where's the DNA polymerase going to add onto? It adds onto the 3 side. So the first DNA is built this way. And remember, that's the lagging strand, because it's the opposite. It gets too far away, so that's another DNA polymerase. But it'll be sitting at the end. This is DNA polymerase 3. The 3 is the one that builds it. So DNA polymerase 3 is building the DNA, but remember, it can't get too far away from the fork, and so it's going to go and get reloaded back on and build, and then reloaded back on and build. So how does it need to build? Always has to have a primer. So you have little primers being made all the time. And then the DNA polymerase comes on them and adds your DNA until it bumps into the next one, the primer that you had before. And then it's going to get another primer here, and then DNA polymerase is going to jump back on here and add your DNA until it gets there, and another primer here, and then it's going to add there. And so in the end, you'll have all these little fragments of DNA that have to get put together. Oops. So this is called an Okazaki fragment, named after the scientist who discovered them. Okazaki fragment here, Okazaki fragment there, Okazaki fragment here. It's part of the lagging strand. And so in the end, you don't want your new DNA to have RNA. You only want it to be DNA. So another enzyme has to come in and to take away that RNA. So we can do this. So this enzyme would be here. Okay, and it's going to take away the RNA and add DNA. Removes the RNA primer and adds the DNA. So anything that's going to add DNA is going to be a DNA polymerase. This is just a different shaped one because it's all about shape. What can enzymes do? Depends on their shape. So DNA polymerase 1 has the shape to where it can remove the primer to get the RNA out of there and add DNA nucleotides. So it'll replace that with the brown DNA nucleotides, so you'll be building a DNA strand. This is good.
So question. So
Yeah.
to build the Ozaki fragment, that's got to be the DNA polymerase 3. Once it builds the fragment, the polymerase 1 comes in and just kind of knocks out the 3.
Yeah, it's gonna do a it's gonna do hydrolysis, break it, it'll pop it off, and then add a new one. Break it, pop it off, and add a new one.
Okay, now where does transcription and translation...
Totally different. So this is all just DNA replication.
Okay.
Yeah. Similar ideas to where it's the complementary base pairing. You have an RNA builder, but in that case, you're only looking at a little part of the genome, and it's not making Okazaki fragments. It doesn't have primase. There are no primers or anything like that. So that's
Okay.
different. That's a different story. Similarities, but different. This is DNA replication only. So what's the issue after that? I don't have it on here, but the issue is now you have your DNA. There's your old DNA. Here's your new DNA, and your DNA polymerase has taken out the primers. This is good, but the way that enzyme is shaped, it just can't add on to this last one here. There's no space for it, basically. So that last little thing. So we call that a nick, which just means it's like one nucleotide that's not sealed. That's not missing a bond. And DNA polymerase 1 can't fix that. So who's going to do that? A different enzyme. That enzyme is going to sit there with the little nick, and its name is ligase, which is an enzyme that's going to ligate. So if you might recall, like sometimes people get their tubes tied, called tubal ligation. Or if you watch CSI shows, they talk about ligature marks. If someone gets strangled, they have ligature marks. So ligase ligature is an enzyme that ties it together. Not literally, but its job is to basically do a dehydration reaction and finally make this last bond. So that DNA ligase ligates or seals the DNA together. Yay, now we have a strand of DNA that's just DNA, and it's all sealed. Yeah, that's good. So the leading strand's pretty simple as far as like helicase unwinds it, topoisomerase is going to keep it from breaking by cutting it and letting it unwind and then resealing it. Single strand binding proteins sit all over the open DNA to keep it from re-bonding to, or re-hydrogen bonding. DNA polymerase builds the new DNA. It has to work off primer, which is built first by primase. This backwards one, the lagging, is a little trickier because you do it in this fragment because it's like build too far away, jump back on, build too far away, jump back on, build too far away, jump back on, build. So you'd have a primase, and there would be a primase here because it's building the new primers that the DNA polymerase is going to come onto. So like primase built this, then DNA poly came on. While that was happening, primase built this, then DNA poly came on. Primase built this, DNA poly came on. So you have the primase keeping on making primers. Then you have the problem of RNA. RNA gets removed here by DNA poly 1. So that's always what's removing the RNA in this case. So primers get removed by DNA poly 1 on both strands. Ligase comes in and seals that last nick. And then you've got a new DNA strand. And in the end, this bubble will go this way. That bubble on the other side will go that way. And then you'll have, in eukaryotes, you have a bunch of them. There's lots of places the DNA starts to be built. And so basically you're building new DNA this way. You're building new DNA that way. And eventually these two, it's hard for me to show you, but these two bubbles will open until they merge. And these two will open until they merge, and then everything will get sealed together. And in the end, you end up with two completely separate new DNA molecules. Each of them is a double helix. And they actually they stay together. I don't want to kill this, but okay, so you have new double helix DNA. One was the parent strand. One was the new strand. You have another DNA double helix. One was the parent strand and one was the new strand. That's these. And they actually stay attached together because they're the chromatids, sister chromatids. So when you talk about, we talked about mitosis and the cells separating, and you talked about meiosis and the cells separating. Like, why in the world are you DNA replicating? It's because you're going to make a new cell and you need DNA to go into it. Because you can't just split up the DNA in the old cell, because then your new cell wouldn't have everything it needs. So this whole shebang here is happening in that S phase, the synthesis phase. So from a while ago, where is DNA replication happening? Here, S phase. So you've got one chromosome, which is your double helix, really, really long. And you have a bunch of them, because cells have more than one chromosome. Those get replicated in S phase. Which is the same thing as like when we draw chromosomes like that. This is the whole thing with the forks, and the forks open it up, make these strands. So really in the end, this is a double strand. This is a double strand, and they're attached together. There's your replicated DNA. And then those are going to go through G2, great, and then they'll get to an M phase, either mitosis or meiosis, and the two new cells will form, and each of them will get one helix. So they line up in the middle, you know, they line up our chromosomes in the middle, they separate in anaphase, and you end up with one in each. Like this. I'll draw them lining up. It doesn't. Yeah. Remember how the spindles are attached? Pulls them apart. So you'll end up with 2 or 4 cells, depending. This mitosis or meiosis would go around again. And so you end up with the chromosome here, which is just the DNA that you doubled, splitting it up, and so in the end you end up back to this, but you just have 2 cells. So you have one chromosome, open it up, DNA replicate with the forks, make 2 chromosomes. Why is that going to happen? Because you're going to make new cells. So you either do mitosis and make 2 cells that are identical, or you do meiosis and you mix it all up and make cells that are unique. And then reproduction can happen. At least in animals, that's what the cells do that are made by meiosis. So where is the DNA replication happening? S phase. Why is it happening? Because you need to copy the DNA before you can make more than one cell, so that every cell gets all the genes it needs.
Okay.
Yeah. In mitosis, in animals, you have a cell that is 2N, meaning it's diploid, meaning it has 2 copies of every chromosomes. So it has 2 big chromosomes, it has 2 little chromosomes, it has 2 wiggly chromosomes. So basically, one was from your mom, one was from your dad. With mitosis, you need the same cells. Let's say you want to make a new skin cell. So what do? You duplicate each of these through DNA replication with the forks. So that's why in S phase you end up with 2 duplicated copies. So you've got duplicated bigs, duplicated littles, duplicated squigglies. Great, then those line up in metaphase, get pulled apart in anaphase, and you end up in both of your new cells having 2 bigs still, 2 littles still, and 2 squigglies still, but it's in both of them. So you end up with the same exact thing where you have two bigs, two littles, two squiggles. So this is just like chromosome number 1, chromosome number 2, chromosome number 3. You know, like in Down syndrome, you have too many copies of chromosome 21. So like that's chromosome 21. Could be, you know, it's just the number of chromosomes. Or this could be 2 X's for a female or 2 Y's. But so this is mitosis. In meiosis, I had an eraser, but cells are starting diploid, so this is in like an ovary or testis. Then they duplicate. Same thing here. Same cells. One from your mom, one from your dad of each of them. This is in an egg or this is in the ovary or testis in animals. There's going to be S phase. The DNA replicates through DNA replication with the forks. And they're not lined up or anything here. Okay, great. So now we have 2 copies of each. And then the cells do the whole thing where they line up with their partners now. Why? Because you only want 1 big in each. You only want 1 little in each. You only want 1 squiggly in each.
Squiggly in each. Yeah. And that's for plant cells, correct?
This is for animal cells making eggs and sperm.
Oh, yeah, that's right.
Metaphase 1. There's the spindle. They line up, paired. Why? Because you want to make sure you sort 1 big into one side and 1 small and big in the other. 1 little into each side, 1 squiggly. So it'd be very hard if you just had them all over the place to like make the cell to make sure it got 1 of each in each new cell. So if you just pair them, it's really easy because it's like, okay, you got a pair of socks. I'm just going to separate them into 2 piles. Versus like, you have the socks all mixed in a basket and you're trying to separate them into each and you're just trying to do it without re-looking at them. You can imagine that's impossible, right? It's like, how would you remember after 20 socks if there was a stripe in that pile or not? But if you just pair them together first, then as you go, it's just like stripe, stripe, you know, argyle, argyle, gray, gray, polka dot, you just separate them. So that's like part of the reason, I mean, that's why they line up. The other reason is that crossing over business where the genes get mixed up. Same genes, they just get fragments. So basically it's like in your body, your mom and your dad's chromosomes exchange pieces. So when you make an offspring, they have mixes of the grandparents. Because, yeah, so it's the chromosomes mixing up bits, but it has to be the same ones. So you only want the big to exchange with the big, not the little and a big, because they don't have the same genes. So it's like the bigs exchange some bits, crossing over in prophase 1. Littles exchange some bits and the squigglies exchange some bits. So then you do anaphase 1 and all that, you end up with 2 cells, and you have a big chromosome in each of them. That was separated here. You have a little chromosome in each of them. You have a squiggly chromosome in each of them, but you can see that you have duplicated chromosomes, right? So we only have 1 copy of each, 1 big, 1 small, 1 squiggle. So that means we call it N. N just means 1 copy of each. So we have 1 copy of each chromosome. But it's duplicated, which is a problem because you have too much DNA. So what does the cell do? It splits it up again. So it would align them in the middle, 1 big, 1 little, 1 squiggle. And this one would align it. And again, this is metaphase 2, because it's only 1 of each. Big, little squiggle. This looks just like mitosis. It separates these, so if we think of this separating, pull 1 big into each cell, pull 1 little into each cell, pull 1 squiggle into each cell, and the same thing here. Put one big into each cell, one little into each cell, one squiggle into each cell. And so then in the end, you end up with 4 cells that are all haploid cells. And then this would turn into sperm or eggs. And why does that? Because when you have a sperm and it fertilizes an egg, you got to get back to your original. So you have one copy of each, a big from mom or from dad, and you have one from mom, then you get baby with a normal amount. You have a little copy from dad, a copy from mom, you get a baby with a normal amount, and then you have a squiggly from dad, a squiggly from mom, you get a normal amount. And so then you'd have 2 copies of every gene, which is good. If you didn't have it, and you just had like a cell combined with another cell, then you'd end up with double the chromosomes in the baby, which should be problematic. And then how does this relate to genetics? This relates to genetics because this is all the big A, little a stuff. This is all this means. What does big A, little a mean? Big A, little a means on this chromosome there's a big A version of a gene, which just means it's the gene with the CGTC sequence. And on this chromosome we have a little a version of the gene, which just means it's the CCTC, so we have a difference here. So it's a heterozygote. It's a difference between the 2 alleles, so that's what this is. It's just like a sequence of DNA. Maybe on this chromosome, it has the B gene in this spot, but it has 2 copies of the same. So in this one we have CCC, and in this one we'd also have CCC at that spot, and so it would be identical, and so that is what is homozygous. So if this one we are doing A's and B's, we'd say it's heterozygous for A, it's homozygous for B, and that's the parent's genotype. So that's where the genotype comes from. That's what it means. It means the sequence of DNA. If it's the same sequence, it's homozygous. If it's different sequences, it's heterozygous. You have 2 of them because you have 1 chromosome from mom and dad. And where did that come from? That came from the eggs and sperm. So really, we have our A here. So let's say that's dad's sperm. Plus mom's egg. So in mom's egg, she has a little a and a big B. In dad's sperm, he has a big A and a big B. When you make baby, you get 1 big A, 1 little a. You get 1 big B, 1 little, or 1 big B, 1 big B, and that's how you get the baby's genotype from the parent.
Now that also be the way that you get recessive and dominant genes?
Yeah, so that's what these mean
A
here.
would be the dominant, little a would be the
Yeah, and I mean, what does dominant recessive mean? Why is it big A, little a? It's because, like, we don't want to write out the sequence every time. Could you imagine writing the difference in the sequences? I mean, it's not always just 1 letter. And so we just came up with a way of abbreviating it by using, like, just single letters. And when, in this sequence here, why is it big A, little a? I'm going to run out of paper. Not really, not right now, but what does that mean? Big A means that this sequence and it's much longer.
Can you touch your screen up just a little bit?
Yep.
There you go.
So let's say big A means this is DNA, and it's much longer.
Yeah.
And it's double-stranded. Big A means this sequence, little a means that sequence. Why is it big A and little a? These are proteins that it codes for. So this sequence here, this codes for an amino acid, right? And if you looked at that codon wheel, you'd start in the middle and you see GT, but it would be mRNA. Right.
We don't, for the exam, will we need to know the codon, like the SER?
I'll give you the wheel. I'll give you the
What was
wheel.
that?
I'll give you the codon wheel and the codon chart.
Okay.
Yeah.
Okay.
So let's say this is the coding strand and this is the template strand. So template strand means that that's what you use to build the RNA. So we matched, this is when transcription would happen. When transcription matched with the G, it put in a C. When it matched with the C, it put in a G. When it matched with the A, it put in a U. So at that spot, this is the mRNA. It has this codon, CGU. There's a lot of other codons, so whatever that one is. So if I look at my codon wheel, I'm going to look at the middle. So CGU, I say CGU as arginine. So this codes for an amino acid arginine. Let's say that's this little arginine. Lid on wrong. Okay. Here. There's the arginine. This was this in the sequence of the protein. That's it here. In this one, we have the template strand being this one. So when it builds an RNA, what's that RNA sequence if it's reading this one? What would it match with a G?
CCG.
So let's look here. CCG is proline. So at that same spot, so there's your proline. That's another amino acid. And it's at the same spot, but let's say it causes it to fold a little differently, the protein. This protein shape does some job. Let's say, what should we do? Let's do like a tail scenario.
Okay, so this is what it was asking on Concord Consortium,
Yep.
where it asked about why do proteins. I kept answering that question the same way. They fold differently, or it asked about the polar and nonpolar
Yep.
molecules in it and
Yep.
why that's important, like how that affects the folding, and this is why.
Yep.
This is what you were looking for?
Okay.
Okay.
So
But I
right,
think
so
that's totally wrong.
something could be different about that that amino acid can affect the folding. So arginine is a, I want to say positive amino acid, because I could give you here. Let me share my screen because this is useful. Hey Justin.
Morning.
Let's see. Share content. Whole thing. Screen. Okay. So there's the codon wheel. There's codon charts. And amino acid. Okay. So here's two things I could give you. All right, so you figured out the sequence because you knew which was the template, and then you figured out what that mRNA complementary to that is. I read my codon wheel here, and that's how we did the CGU. So we start in the middle, CGU, arginine. That's how I knew what amino acid was there. And then you see the different one is CC. Oh dear, look what I did. That's bad.
You have to say.
Yeah, so let's do this and make this. Two changes. You could have that. So CGT, CCG. This is the same codon in there. I mean, it's a different code, but it's the same part of the DNA. So it's just two nucleotides are different. In this case, the two nucleotides being different codes for a different codon, which codes for a different amino acid. So that's proline. And then there's your proline. How did I CCG? Yep. Now let's look here and let's see if there's a difference. So we started with arginine, positively charged. Oh, I think I got that right. Okay, positive charge here. So that was arginine. Let's look at what proline looks like. Proline, you can see has lots of CHs. So if you remember from very beginning of the semester, CHs are nonpolar. So I would expect you to know that. So CHs are nonpolar. So now we have a nonpolar amino acid versus a positive amino acid, and this could cause folding differences. It could cause bonding differences. It might bond something more, it might bond something less. It could cause nothing to happen different. Even if it's a different amino acid, it might not change anything that matters. And so that's why you get different phenotypes in the end. Let's say this one codes for 5 backbone vertebrae, or tail. Let's say tail. This one codes for 5 tail vertebrae, as in when it helps build, when that enzyme helps build bone, it ends up with 5 vertebrae. This one doesn't work quite as well, or maybe it just stops sooner, like it responds to signals to stop. So this enzyme only ends up with 2 tail vertebrae. Call it a short tail, let's say. Now, of course, this organism, because this is just one individual, let's just call him Drake. This is just one Drake that has both of these. So is it going to have a long tail, a short tail, or a kink tail, or whatever? It depends on how these phenotypes not really interact, but what do they do to the organism? So if there's a situation where having an arginine here with the 5 vertebrae or 5 tail vertebrae versus 2, maybe that ends up giving you, the way that the enzymes work, maybe it ends up giving you 3 vertebrae. So in the end, your organism would have a middle-length tail. So that would be one of those incomplete dominants where a big A and a little a gives you an in-between. That's one possibility. What else might be the case? Maybe instead of this being the result, maybe when you have this protein here, it does a really good job and it makes all the tail vertebrae, even if there's only one version of it. Like this one's not doing as good a job. This one's doing a good job, but maybe it's enough. Maybe it's enough. So instead of getting an in-between tail, maybe when you have the combo here, you get the long tail. Sorry, this is all over each other, but maybe when you have these, this is enough to get you 5 vertebrae. So even though there's this one here that's only working like a little bit, this one's enough to get you 5. So then that would mean big A, little a gives you big A phenotype. That's complete dominance. Now this relationship here is always going to be the same. It's not like half the time it's going to be completely dominant and half incomplete. It won't be like that. I'm just giving you different scenarios to where different combinations of proteins may give you different results. So if it did this in the end, and where this combo here, which in the end means it's a mixed combination of sequences, if that gave you a long tail, that would be called complete dominance, where A is completely dominant over little a. If it gave you a scenario here where when you had the mixed genotype and you had half the protein working a little bit, half of it working a lot, you ended up with a middle tail, then that would be incomplete dominance. And then what is all the Punnett square business? The Punnett square business is saying if this drake had babies, what's the probability of inheriting this gene that gives a proline here? What's the probability of inheriting this version of the gene, which gives you this arginine here? And you can see that the drake would give one allele to its baby. So half the time, you expect half the time it would give this version of this gene to the baby. Half the time it would give this version of the gene to the baby. And that's why in a Punnett square, you do the business like this. This is for dad up here. Half the time you give the A, half the time you give the little a, because half the sperm have one, half the sperm have the other.
And that's just totally random. There's no...
Right. Which one it gets? Yep.
Yeah.
And then how do you know what the other combo it is? Depends what mom is. So if the mom is...
This is by chance.
So let's say mom is big A, that means all her genes have, all her versions of this gene have this sequence. So all of her versions of the gene all make the arginine version of the protein. So what's mom going to give baby? Mom only has one option. Like, she's going to give the baby this sequence that gives you this protein. So the only option for mom, all her eggs will have the same exact thing, but only has one copy because you don't want babies with 4 copies of each gene. And that's why do we only put 1 here instead of 2? That's because this is an egg. And eggs are formed by meiosis, which, as we saw, gets you half the amount of chromosomes because you're going to put them back together. So that's when this combo happens. So what's the combo? This Punnett square, all this means is that what happens if the baby gets the sperm that has this sequence, and then it would get the mother sequence. You get an offspring that has big A, just meaning that it has, both genes are this sequence here. So baby has 2 copies of this, which means baby makes 2 copies of that, which means baby gets long tail.
Gotcha.
But you might have a sperm with this version in it. That's what this option is. So what happens there? Well, if you get this version of the gene in the baby, and then mom still only has the same one, so she'd still get the same one. What does that mean? That means that you get a mix. And what does that result in? Depends on the inheritance, but let's say it's incomplete dominance, or let's say it's complete dominance. So this scenario here, where you have 1 of each, you still get a long tail, because this one works enough to make a long tail. That would mean that you get all long tail babies, because these are giving you long tails. This combo is giving you long tails, and so you get 100% long tails.
Gotcha. Okay.
Or let's say it's not this scenario. Let's say it's this scenario where you get the in-between. So when you have a long tail, you know, a protein that makes 5 vertebrae and a protein that makes 2 vertebrae, let's say you get a baby with 3. So in this case, if your baby ended up with this sequence from dad and that sequence from mom, all its proteins are the functioning type, then you get 5 vertebrae in that offspring. In this one, if you have 1 of each and it's incomplete dominance, then you get the 3 vertebrae scenario. And that's why if I ask you, what's the probability of getting 3 vertebrae? You'd be like, oh, there's 2 options here. Only one of them gives you that, 1/2. What's the probability, or what's the ratio? What's the genotypic ratio of the offspring? The ratio is just however many you have of each. So 1 big A for every that one, you expect 1 big A little a. Now, does this mean it'll happen? Not necessarily, but that's the expected genotypic ratio.
But there's, I mean, you can do the ratios and get a percentage of probability.
Yeah, that's what this is.
This is obviously no exact.
Well, that's what probability is, right? It's a likelihood. Now, if you had a fruit, like let's say this is a worm, and the worm, or fruit flies make a lot of offspring. So let's say it's a fruit fly, and it had like 1,000 offspring. You would expect a pretty close ratio of 1 5 vertebrae for every, like 50% or 1/2 of them would be 5 vertebrae, half the offspring would be 3. You'd expect pretty close to that because you have like 1,000 offspring. But when you only have 3 offspring, then, I mean, or 4 offspring, are you going to get 2 with 5 vertebrate tails, 2 with 3 vertebrates? No, not necessarily. Let's say you have 2 babies. Is one going to be this and the other one going to be that? Not necessarily. It's just the probability is half and half each time. That's why, like, some families have 5 girls as kids, and they're like, we should have another baby. We're due for a boy. But that's a fallacy because every time it's still half and half. And even though it feels weird, remember, this is like a good life idea. The life idea is that even though it feels weird for statistics to happen to you, like, what's the probability of us getting hit by Harvey in Houston and then 2 years later getting this COVID-19? What's the probability of that? Low.
Yeah.
But it happens. So it feels weird when it happens to you, but if you look in the scheme of either like the entire population, like time scale, you know, looking at billions of years of time, what's the probability this would happen in billions of years of time? Pretty high. I mean, it's got to happen to somebody. You know what I mean? You're the family with, 6 boys, and you're like, how can I have 6 boys and no girls? It feels weird to you as an individual, but probability says this happens to somebody. Even if it's a 1 in a million chance or whatever like that, that means still 1 in a million people, and you are that person perhaps. So even though things feel funny sometimes, it's just because we're looking at it from our own perspective as one person instead of looking at probability here. But if you had a lot of offspring, you would expect that you'd have 1 3 vertebrate for every 1 5 vertebrate. And we could call this 1 long tail to 1 medium tail.
Do the screen unshare so I can see a bigger picture.
Sorry, I forgot that it does that.
That's right. Okay.
Oh, it's all messy.
That's all right. I mean, I was trying to watch it, and I think I kind of understand it. I mean, I'm sleepy, so I'm just focusing as hard as I can. It makes sense. Now, whether or not I can repeat exactly what you're saying verbatim.
Yeah, no, I mean, and what I want, I mean, I'm going to do for the exam, I need to do, like, one of our learning outcomes is working genetics problems. So we have to do some of that stuff. But for me, and the reason I have everyone doing GenAventure is because it's more important for me that you understand the concept of, like, what does a big A, little a mean? No, I'll tell you what, I would say 80, if not more % of students go through maybe all of biology, even get a degree in it, and don't actually understand what the heck a big A means. They just know to put it in the Punnett square and count the number of boxes, which is useful. But in reality, is a Punnett square that useful? I mean, we have simple probabilities of, like, what's your probability of having a baby with cystic fibrosis if you're a carrier, meaning a heterozygote? That's useful. But really today, the kind of genetics we care about and the kind that affects most people is kind of complicated stuff where it's a lot of factors, where it's like, probability, right? What's the probability? Okay, you have the BRCA1 mutation. That's the breast cancer increase. Are you going to get breast cancer? Increases your probability. What else depends on? It depends on environment and how that interacts with the genes. It depends on your other genes and how they interact, right? So in the end, what matters is that you understand what it means for probability, and that you understand that the combination and the outcome is from a bunch of genes most of the time, and how they all make proteins or regulatory molecules, and how that in the end shapes what the thing looks like in the end. Because, like, let's say we can do a problem that's a little more complicated. And this, unlike DNA replication, which is just making an exact copy of the DNA, which does happen in your S phase.
Yeah.
For both mitosis and meiosis. Mitosis in animals is going to make body cells. Meiosis is going to make eventually eggs and sperm. All this business here, this is the eggs and sperm from meiosis. This is transcription translation. So we've got transcription, reading the DNA to make RNA. Translation, reading the RNA to figure out the amino acids. And then protein folding. So that's transcription here, translation there, protein folding after that. And then we work Punnett squares, basically to figure out with your combination of DNA sequences, what's the outcome. So let's look at something like combination where, so let's just look at 2 genes and the proteins and why it would be a result like it is. Okay, so what if we have cats? This is how this actually happens. So is that better?
Yep.
Okay, so we've got 2 W, we're going to call them W. What does W mean? This means you have a DNA sequence that gives you a protein. Big W means you have a DNA sequence that is going to block pigment transport in hair of cats, the fur.
Okay.
Let's just do this. Let's say your little w stands for a DNA sequence that gets an early stop codon. So when you're reading that DNA, when you're reading that codon wheel, you would read the codon and it would say stop.
Okay.
So it ends up making a really tiny peptide that doesn't fold, so you end up getting no protein. It won't fold. That's what these things mean.
Okay.
Okay. Then let's look at a separate gene. Just trying to think if we want to go crazy and do X-linked.
Let's kiss it.
Try it or do something simpler first.
Something a little bit simpler.
Okay.
So, I guess if you have a protein that blocks the pigmentation, and then on the opposite side, just like you have, it has no protein. That's going to give you a more likelihood of a light-colored mouse or a light-colored cat.
It depends on which cats you're crossing, right?
Yeah.
If this is, so this would be a complete, well, depends on how it works. But let's say, we have to tell you this. So let's say when you have so big W is completely dominant to little w, meaning if you have any protein that blocks the pigment transport, in this case, you won't get any pigment. So in this case, big W is going to give you a white cat. So if you have a big W at all, it's going to have the blocking protein. It's going to block that pigment from moving, even if it's there. Even if you have pigment, it's not going to matter because it doesn't get to the hair. So whenever you have any protein that blocks it, you get a white cat. So that's why we call it big W, because that's dominant.
Okay.
In this case, you have to have little w's where you don't have any blocking protein, so no blocking protein. So if there's no blocking protein, then you get color cat.
Okay.
How are you going to know what color you get? Depends on the other genes that are there. So let's say, and this is part of cat color. This is all true. So let's say you have we'll do an easy one. Okay, let's say you have, this is that dark pigment. So this is a little hand wavy of how this works, but I'll explain in a second. Okay. So eumelanin is a molecule that reflects light to where it looks dark brown, black. Dark brown, black pigment.
Okay.
So big B just means that you code for a gene that codes for some sort of protein that gives you like a round cluster of eumelanin. In the end, it builds round clusters. Little b means you have another version, same gene, but you have a different version, a different sequence that codes that ends up giving you triangle-shaped clusters of eumelanin.
Okay.
So when you have round clusters, what humans see, what's reflected, gives you very dark. A lot of eumelanin, it reflects and it makes it look black. So a cat that has round clusters looks black.
Okay.
When it is reflecting from a triangle cluster, that looks brown. And we'll say that having any round clusters makes the cat look black, which just means that big B is black, little b is brown because black is dominant to brown. In this case, other cases it wouldn't be like this. Okay, so that's our setup. Great. So these are important to write this out so you don't get confused about what's happening. So your white is dominant to colored and your black is dominant to brown. In this case, both complete dominance. We'll do another one in a second. Can you still see that, the words?
I can see the whole thing now.
Okay. So let's say we have let's just go for it. We have a white cat with a brown father and a black cat with a brown mother, and we want to know what's the expected phenotypic ratio of the babies? So first thing you have to do is figure out what that means genetically. So we know that having a white cat, how do you get to be white?
The white has to have the prominent white or W.
Yep, the big W for dominant. So basically it has the blocking pigment, which means it has a big W.
Yep.
And then
the brown father is going to be the small b.
I'll just do this because we're looking for two genes. So dad has to have it to look brown. He has to be two little b's to look brown. If he had any big b's, he'd look black. Yep. What does he have to be colored?
Well, for color, he's going to have a black cat is going to have the black dominant gene underneath the white.
So let's continue going with the father first. You have a brown, but how come he's not a white cat?
Because he's got the U. Was it the U?
Little W.
Yeah.
That's my little W with a line, so I can distinguish it from my big one. Yes, so basically he has two copies of the gene that don't make any blocking protein, which allows the color to get transported to the hair. And then when he gets transported to the hair, it's the triangle clusters, which makes the cat look brown. So in order to look brown and not white, the dad had to be little w, little w. So we know that.
Yep.
So what does that mean for our white cat offspring? I can tell you some other stuff. Let's see. Okay, that doesn't matter. Okay, this is all you need to know. Okay. I gotta tell you, this white cat also has a black mother. Oh, but they can't be because the house is gonna have a white baby. Has to have a white-coated mother, but there, I'll tell you that. Mom's genotype for the brown-black allele, the eumelanin allele, is big B, big B. She has to also have a big W because where did that come from in the baby? Had to come from mom. So mom has to be a white cat, but she has the two big B's. I'll just have to tell you that so you can know what's in the baby. Okay, so mom, we know the white cat because it has a coat. So what did it get from dad? It only had one option to get from dad, right?
One small W.
So dad had to give it one little W, so it has to be heterozygous for that. Then what did mom have to give it?
Two big B's.
And then, just one, right? Because mom has only big B's, but her eggs would only give one to the offspring, and dad only has little B's, so he'd give a sperm to the offspring.
Yeah,
right.
I got you.
Now, I could give you information about this, like the brown father. We didn't need to know anything about its parents. So you don't have to have parent information. It just depends on the inheritance of what I tell you. So like sometimes you'd be able to figure this out all by itself. Sometimes I have to give you info. Okay, so that's the parent here. This is the black cat with the brown mother. So let's figure out what that parent is. Then we're going to cross those. Let's see. So we know we want a black cat. So how does it get to be colored and not white?
It's got to have, it can't have that blocking protein.
Right, no blocking protein.
It has to have a small W.
Yep, so it can't have any blocking protein, which means it needs a sequence that we call little w. Yep. And then in order to be a black cat, you have to have
the big B.
Mm-hmm. So it has to have at least one big B, because that gives you some round clusters. The second one could be a big B again, or could be a little b, because it still looks, but you know, because it has a brown mother, that meant the mother had to be little b, because she's brown. That's the only way to get brown. So that means that you have this cat. Okay, so those are our two parent cats. I might also give you some info, like, here's the two genotypes. What's the offspring? Might be like, less crazy. So what I would do is 2 Punnett squares. And let's just see the options. What's the options from the white cat? Let's say that's the mom. So the white cat, what kind of W can it give the baby? So it can give the baby a big W, meaning it has the, would give it the blocking protein.
Yeah.
Or the other half of the eggs would have the little W sequence, which gives you no blocking protein. Okay. And what about this black cat parent? Black cat
is going to give it one big B and one small b.
Do W's
and
first. Just the W's first.
Oh. So from the... You still have to give a big B.
Don't worry about the big B's, just the W's.
Oh, so you got 2 small W's there that you just overlapped?
Yep. So this is a little W, this is a little W.
Okay, so you're going to have another?
So the black cat, this black cat parent, only has little W's to give. All its genes are the same sequence. All of them are little W's. So there's only one option for that cat to give. So let's say this is mom and this is dad. So the mom black cat, her eggs, half of them are going to have the big W with the blocking protein code. Half of them have the little W with no blocking protein. This dad cat, the reason he's black is because he has no blocking protein at all. And so he can only give a non-blocking protein allele to the offspring. And you got to work the W's together and the B's together. You can't mix W's and B's together unless you're doing doubles, but I would do it like this. Okay. So what's the offspring possibilities? You could have a baby that has a big W from its mom and always the little W from its dad. Or you could have a baby that got the little W from mom and then always the little W from dad. So those are our 2 options for baby W sequences. Now we can go to the B's. So let's not worry about this right now, but let's go to the B's.
Okay.
All right, so we just look at the B's now. So what's the possibilities? And you'll notice here I didn't draw another little W because it would be exactly the same thing twice. That's a waste of our time. We don't have to draw another little W again because it's a replicate. So here, what kind of B can mom white cat give the baby? Half of her B's are the big B.
It has to be a big B.
So half her eggs should have a big B option for the round clusters, but then the other half of her eggs have the little b for the triangle clusters. So half of her eggs have each, and dad has, when we look at his B, half of his sperm should have the big B for round clusters, and half of his sperm should have the little b for triangle clusters. So if we look at these options,
little b, big B.
So all I'm doing here is doing egg sperm combos. Egg sperm combo 1, egg sperm combo 2, egg sperm combo 3, egg sperm combo 4. So those are the possibilities. Here we only had 2 egg sperm combos. Why? Because it's homozygous. Okay, so here we have 4 in this case. So now I want to know what the phenotypic ratio of babies is. So what I would do here is I'd write down what color babies I can have. So based off of what I've given you, what are the 3 kind of colors of babies you can have?
You get the left one with the...
Don't worry about this yet. Don't worry about this yet. Just in general, what kind of offspring can I get?
It's the ratio is going to be...
Don't worry about the ratio.
1-1.
Don't worry about the ratio yet. Just what kind of cat colors can I get? What are the 3 cat colors that we're coming up with? Period.
White, brown, or black.
Good. That's it. So we have a white cat, we have a brown cat, we have a black cat. And what I like to do is to say, what genotypes do I need to get each of those? So each of them is going to have W's and B's. So I write this down so I don't get confused. So I know I need 2 W's and 2 B's in each. And the question is, how do I get those? So how do you get a white cat? You need the blocking protein.
Yep.
So you need a big W. And that's blocking transport, so no matter what else you have, you block the transport, so it doesn't matter and you look white. How do you get to be a brown cat? First of all...
Anything else in that?
Yep, anything else would give you a white cat because you blocked the pigment. To get a black cat or a brown cat, so to get a brown cat, you have to get the color first, the transport of the pigment. So that means you have to have no blocking protein, which is little w, little w. Then, in order to get the brown color, you need triangle clusters.
Yep.
So to get only triangle clusters, that's what we call the little b's. And we know that here because it's brown. So in order to be a brown cat, you have to have this combination.
Small b's.
To be a black cat, you have to have color again, so you can't have any blocking protein. And you need some round clusters. So you'd have to have round cluster gene. And then because round clusters, even if you just have some of them, it gives you a black cat, that's all we have to have. So having all round clusters makes a black cat, and having combo makes a black cat. How do you know that? Because we said it's completely dominant.
Okay.
So then you can just look at here and see what the probability of each of these is happening. So what's the probability of getting a big W? So you go to your W Punnett square. What's the probability of getting at least one big W? So which of our boxes have
25%.
Where's your 25?
Well, inside your block, you got your capital W inside the left,
Mm-hmm.
and small three small W's. So 1 out of 4.
How many options do I have here? What are the two
Oh,
genotypes?
2. Okay, so 50%. I see what you're doing.
So there's only 2 options for babies in this case. So we have the probability of getting at least one big W is only this. And we have the probability of that happening. There's only 2 options, so that's half the time. So we have 1/2 times anything else. Doesn't matter what B's there are. So our total probability of getting a white cat, and we'll put it in a ratio, is 1/2 right now. Then we go, what's the probability of getting 2 little W's to get you a colored cat? We have 2 options.
2 options, yeah. So 100%, basically.
Why? What are our
For
2
that
options
side. Well,
here?
you get them on both sides.
We do have them. So we need 2 little W's.
Okay, yeah, if you have 2 little W's, 1 from each side, that's 100%.
But we only need 2 of them. So this one's a big and little, right? That option.
Okay, so then you're back to 50%, or on this side,
Yep,
yes,
50%. Yep. So all we look at is how many boxes fulfill 2 little W's. We have 2 options, and one of those boxes is 2 little W's.
Okay.
So
That's
the
what
probability
you
of the baby getting 2 little W's is 1/2, because half of them get that, half of them, well, the probability is half of this, half of that. So the probability of getting 2 little W's is 1/2. We look in here, what's the probability of getting 2 little B's? So how many of our boxes have 2 little B's?
3 of them, so 3/4.
2 little B's in the same box.
On the same box, that's 25%.
Yep, just this one. This is the only one that has 2 little B's and just 2 little B's, right? So we want 2 little B's to match this. So that is 1 out of 4. And then you multiply because you need both that and this. So 1 times 1 is 1, 2 times 4 is 8. And then to get a black cat, we need colored. So 2 little W's. So what's the probability of getting colored cat? Just that one, which is half again.
Yep.
Times the probability of getting at least 1 big B. So how many of these squares have at least 1 big B?
1 big B is
At least
3.
So at least 1 big B, at least 1 big B, at least 1 big B. So there's 3 options to get at least 1 big B. So that's 3 out of 4 options, and that gives you 1 times 3 is 3, 2 times 4 is 8. And now to put it in a ratio, we've got to make it all have the same denominator. So 8, 8. So let's turn this into 8. So how do you get to an 8 on the bottom? You multiply 2 by 4, and that would give you 4/8. So in the end, what's my expected cat baby ratio? I expect 4 white cats to 1 brown cat to 3 black cats. Yep.
Okay.
could also say, what's the probability of getting a white cat? And then you just look here and you'd be like, oh, 1/2. Or I could ask, and so you wouldn't even, if I just asked about white cats, don't do this. You don't need to do that and waste your time. Just do whatever I ask you for. But in this case, I ask you for all of them. And you don't write 0 of anything. There's no such thing as a white-brown cat. Right? So sometimes people want to say white and brown together, but there is no such thing as a white-brown cat. It's white, brown, or black.
Brown or black.
So this is where we're looking at well, it's a hybrid, but we're looking at 2 genes giving you 1 outcome. So it's called a polygenic trait, where you have a trait that is from more than 1 gene. Poly meaning many genes. Polygenic.
Okay.
So you just have break it down with the W's, break it down with the B's, break it down what combos, and don't get freaked out. But in the end, you can see what this actually means. This means a cat who's white, that means it inherited a blocking protein from its mom, and so it was a white cat. If it's brown, that means it inherited no blocking proteins from each, and it inherited 2 triangle cluster pigment, and so that made it brown. If it's black, then it inherited again no blocking protein and at least 1 round pigment cluster, and so gave it a black cat.
Okay.
And again, why are these like this? This is the results of meiosis. Half of the splits in meiosis gave a big B in the mom. Half of the splits in meiosis gave a little b. So half her eggs are this, half her eggs are that. Half her eggs have this, half her eggs have that.
And which egg we get is random.
Which one you get? Which ones combine together? Exactly. That's why we do equal distributions here of the combos. Now, what if W and B were on the same chromosome? This is where we call it linked genes, not X-linked, but linked genes, meaning if you have them on the same chromosome, especially if they're close together. Let's say this is the chromosomes here. That's 1, and then she has 2 chromosomes, right? Because she's diploid. Let's say, for example, the white cat had on her chromosomes, she has a big W and a big B and a little w, little b.
Tip up a little bit.
Sorry. So what if on her chromosomes in her cells, this is her cell, this is her ovary cells, all her body cells, she has the big W and big B on 1 chromosome and the little W and a little b on the other. This is problematic because we can't do this Punnett square business because big W and big B are traveling together. So if the offspring inherits a big W, because the genes literally are physically together, it's probably inheriting a big B. What we're doing here is saying that there's an equal likelihood of inheriting a W with either one. So we're saying, okay, W's do their own thing, B do their own thing. But when they're on the same chromosomes, it's different. So you can't use Punnett squares. I would never ask you about probability in a linked situation, but you do need to understand what linked means. Linked means that you're not getting these numbers you expect. The genes aren't mixing like you'd expect it. So if they were on the same chromosome, in this case, this is not the scenario. Well, let's not do B. Let's do C, because I don't want you to confuse you. Okay, C gene is something different. C gene is for, I don't know, long hair, short hair.
Yeah.
Okay, so we have these together. And so we couldn't work Punnett squares with W's and C's because big W, big C is inherited together most of the time. And little w, little c is inherited together. So it's not all mixed up because they're literally, physically together on the same chromosome. So the way you would tell that is that you look at offspring and they're not getting the expected mixes. So like you said, it should be an equal probability of all of the different combos. But if you're looking at your ratios and they're really crazy, you barely ever get certain combinations, especially if you get tons and tons of white cat, well, not white, white cats with short hair. Let's say big C is short hair. White cats with short hair, and you have a lot more of those than you'd expect. Then you're thinking maybe white and short hair are linked together because I'm getting these combinations way more often than I should. So maybe the genes are traveling together. So that's a linked gene. So I would not ask you to order linked genes or anything like that, but I do need you to understand that they can't do Punnett squares with those on the same chromosome. They travel together.
Okay.
You'll notice this is a 2 cross, and like some people are like a 9 to 3 to 3 to 1. If you've done the dihybrid crosses and you did this, if you remember that, big A, little a, big little b, big A, little a, big B, little b, dihybrid cross, and you got the 9 to 3 to 3 to 1 ratio, and you're like, well, why don't I get 9 to 3 to 3 to 1 here? It's because you, in this case, A's do their own thing and B's do their own thing. So like, for example, in the examples in the book, it's like you have tall versus short pea plants and you have green versus yellow pea plants. So both of those are not affecting the same phenotype. Short, tall is its one thing. Color of the peas is its other thing. Here, though, it's all affecting coat color, right? You have 2 genes, but it's both affecting coat color. It's not long hair, short hair, and white, black. Yeah. It's all affecting whether it's white, black, or brown. So that's also why these ratios come out different. So I just don't want people to be lulled into thinking, oh, it's always 9 to 3, 3 to 1, or 1 to 2 to 1, or 3 to 1. It just depends on inheritance. And what does the ratios come out like? It just depends on how the proteins or regulation behaves.
Okay.
Because then it's possible we have a situation like where you have like in between stuff. So instead of, how do I do this? So instead of just a black cat or a brown cat, there could be a combination where it's like you can get a black cat, you can get a gray cat, or you can get a white. I'm going to say white because it's a different white. Let's say there's a combination where you could get a long-haired cat, a medium-haired cat, and a short-haired cat, or a long-haired cat, a short-haired cat, and a no-haired cat, something like that. Why is that different? That's different because whatever proteins it's making behave differently. So, or maybe regulation. Let's say the long-haired cat has regulatory sequences that allow full expression of the protein. Proteins fully expressed in both alleles, get a lot of protein, get really long hair. Let's say the combination of having one gene that allows expression and the other gene that clamps down on expression. So there's one sequence that's full expression, there's one sequence that's partial expression. Let's say that gets you a medium-haired cat. And then let's say you have 2 of the regulatory sequences that allow no expression, then you get a no-haired cat. So so that's incomplete dominance, where you have like an in-between phenotype, no hair, long hair, medium hair. And why is that? That's just because the proteins behave differently. And why is that? Just because they're different shapes. Or in the case I'm just talking about, the regulatory regions, like where transcription factors bind. So in chapter 15, they talk about transcription factors, very important. They're basically proteins that turn genes on and off. And they're very specific for different genes. And most of the time, genes need a combo. So this is how your cells are different. So like your eye cell, your lens cell, and your skin cell, and your liver cell, and your, I don't know what else, heart cells, all have the same exact DNA in them. Why are they different? Because different genes are on and off. And why is that? Because different transcription factors, which are just proteins that turn genes on and off, are in combos. So like if you have A, B, and C genes turned on in one cell, that gives you a lens cell. If you have A, B, and F genes turned on, that gives you a liver cell. If you have B, C, and Q genes turned on, that gives you a skin cell. So it's and why is that? Just because those different proteins are expressed. So it's like you turn on some genes to turn on other genes to make proteins that build stuff and give you body parts. Or they build enzymes that do reactions. like that. So it's that chains of expression. And how do you get the first expression? Mom's egg has initial transcription factors in it that start turning on and off genes, which then cascade. And they turn on and off genes in different areas of the egg and so forth, and it's just this cascade.
Okay. It makes sense. Yeah, it looks like we got about 10 minutes left. So for the test, I mean, I've got a really good working idea of this. But can we do kind of an overview of? I know you can't tell me the questions, but
so I did post all the. Just put my table back for the moment. I don't know. I guess it's the floor. Okay. So I can sit down. Whew. I'm losing my voice. I got out of practice. I was like, oh my gosh, when I have to go back and teach and stand for 6 hours in a row. Yeah.
your feet are going to be burning.
You really have to get in shape for that. It sounds stupid, but it's true. If you lose that real quick. But so like my voice. Apparently, people who go to I don't know what they call, ENT people for throat stuff, specifically ontologists. I don't know. But anyway, they're mostly people that are vocalists and sing and teachers. This is what my sister told me. So, okay, what did I post for you? I posted, I just compiled all those learning outcomes for share from the different modules. And I'll just double check and look at them to make sure I have everything I wanted. So I put this up this morning. I don't know if you saw it or not, but you had asked, answer key points of study. So I just took all the learning outcomes from the different modules, put them all together in one spot so you can see the different chapters. And so that may help you have them all there together.
Okay.
I often, during these units, I usually would give a reading quiz at the beginning of every class. And that's good because a lot of times I focus on things that I know you need to do for the exam or really important ideas. But I didn't want to give you guys more things that you had to do for grades. I know that there's enough stuff already. But I thought, it occurred to me that it might be useful for you to see what those quizzes were so that you know kind of things that I would make sure everybody understood. So I think I'm going to, maybe on paper, but I'll take some pictures and put this stuff up. So you can see. Like, for example, one of my quizzes is this guy. It's a DNA replication fork, and I was like, label all the different parts. And then we went over it so that you would know. And I mean, we went over it, but I mean, I'd go over it in class, and then you'd know, oh, that's something she thinks is important. Or have, like, this is all DNA replication stuff, but here's another, it's a quiz. No, this is my notes. But that's one of them. I got stuff that's pictures of other processes, some practice problems. And like I mentioned to you and Abby earlier, I hadn't had the recording on yet, but like I mentioned, some of the stuff is like mastering. Some of the stuff is like concepts from the Geniverse and the different modules and all that stuff. Of course, it's all the same content, but I'm pulling from both those sources. And then I'm pulling from that HHMI, like the DNA discovery and structure of DNA that I had you do like a worksheet on a quiz thing about Watson and Crick and Rosalind Franklin and the DNA double helix and stuff. So you can go back and the answers are all up about that. So go back and look at the stuff I've had you do. because I'm
Okay.
specifically targeting how that material is presented and that kind of thing, because I want it to be in alignment with, what I'm asking you to know. So these
Okay.
learning objectives are also useful. So I'll do that
Yeah,
with some quizzes.
I think I already did the video of how they found the double helix.
Yes, I think everybody, almost everybody, if not everybody, had that. So I have practice problems up for genetics.
You have practice problems up? Where are those at?
In the module. So there's a thing that's called How to Learn Genetics, and it has everything linked to it, which is pretty thorough. So
in chapter
11, 12. So we have a thing here where it's like guide to solving, some practice problems, my video of how to work those, really simple ones, but example karyotypes, here's some different packets and the keys. I didn't have you actually do these this semester, but you can, because we were busy with other stuff, but those are good places to go to practice word problems, and then you can see the results. And I have like how I work the problems on here too, so that you know how I thought about that. So that's one spot. So yeah, just don't forget about DNA replication, because sometimes people struggle with that some. So here's that. Let's see.
So is there something that you would recommend? Yeah, obviously I'm struggling for time. What you have more of, what you have less of.
I try to equally distribute between the chapters. I would say chapter 15, I've kind of really simplified stuff, and what I need you to know for 15, I pretty much cover it's definitely in the lectures or the lecture slides of what you need to know, and then I talk a lot about it in the modules, chapter 14, 15 lecture on there. So basically, you don't need to know super details about operons, but you just need to know generally it's like it's in bacteria, and it's coordinating a bunch of genes. They are all expressed together, and then it's transcribed all as one, and then translated in chunks. But I don't ask too many details about operons. So
Okay.
that's something in 15. So in 15, like I'd say that's more limited than the other chapters. 14 is very transcription, translation, regulation of that, pretty detailed. This idea of mutations and how that has consequences. Different types of mutations. I did write up this thing. Not sure if everyone saw it. Let's see. The guide, yes. Okay, so when you click on the assignment for the How Do Cells Make Proteins thing, there is this study guide and outline. So the Concord Consortium is pretty straightforward with the practice of transcribing and translating, but I did write this up to go along with it. And I mean, a lot of it's like, look at this part of what's happening. Look at this part of what's happening. What does this mean? Here's the different mutations, that kind of thing. Here's how you'd read the sequence. Here's the, like practice doing the transcription translation, that kind of thing. What's a silent mutation? So anyways, if people didn't see that, that goes along with that one about, this has good stuff. And I do have, I'll finish them today, comments for all these. But, it's fairly simple. So I wanted to make another thing for you guys to like kind of get into it. So I had linked to that on there if you hadn't seen it, the guide for that.